You're at the cutting edge of RNA Therapeutics. I'm your host, John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist in the Texas Medical Center. I'm also the director of the Center for RNA Therapeutics, and one of the faculty in that program is sitting to my left, Dr. Ewan McCrae. Dr. McCrae is an assistant professor in our Center for RNA Therapeutics, and today he'll be introducing our guest, Dr. Riju Das.
Thanks, John. It's my great pleasure to introduce Professor Riju Das. Although his accolade list is rather long, I feel like I'd be remiss not to mention that Riju was born here in Houston, Texas. He got his undergraduate degree in physics at Harvard, following which he got a Marshall Scholarship, which brought him across the pond to the UK, where he did a master's in experimental cosmology, molecular phylogenetics at the University of Cambridge and University of College London. Following that, he came back to the United States, where he did his PhD at Stanford, again in physics, with Sebastian Doniak and Dan Hirschlag. Here is where he really started working on RNA, studying the visualizing forces and fields of self-folding RNA molecules. Following that, he did a postdoc with David Baker in Washington State, where he really began developing algorithms for predicting RNA structures, as well as these really cool high-throughput contact mapping approaches, which I believe we'll be hearing more about today. Mm-hmm. After his postdoc, he started in Stanford in 2006, I believe, as an assistant professor, continuing the same sort of work on RNA structure predictions. He quickly rose through the ranks, getting many awards, a Burroughs Wellcome Award. He's a Stanford Medicine endowed faculty member, full professor in 2016, selected as a Howard Hughes medical investigator in 2021, the same year he founded his RNA design company, Inceptive. And yeah, Riju is well renowned in the field for empowering citizen scientists through this massively open laboratory, Turna, where everyday people can work on the RNA folding problem. Mm-hmm. And he's done similar hugely collaborative efforts with things like the RNA Puzzles contest, critical assessment of structure prediction contests involving RNA, and more recently this Kaggle competition to develop new algorithms for RNA structure prediction. Mm-hmm. So I'm really excited to hear what Riju has to talk about today. And yeah.
Wow, can't wait to hear about it. Riju, thanks for joining us on the show, and really looking forward to hearing what you have to say about RNA folding, RNA structure. Maybe first though, you could tell us a little bit about your laboratory, where you are right now, and a little bit about how you got to where you are now.
Yeah, well, first of all, I just want to thank you for the invitation to come and be a part of CV Live, and it's really good to see both of you again. I was visiting Houston earlier this year, and I got a chance to see both of you. And also, as Ewan mentioned, I was born not too far from where you are right now. But right now I am in California at Stanford University, where my lab is. My lab is, we're just a few people, maybe about 10 people. We're really focusing on the basic science of how RNAs fold and how they function in modern living cells and perhaps at the origin of life. And then we're also very curious about how they might impact the future of the world through things like RNA medicines. And what I love about the lab is there are a lot of people who, like me, jumped into biochemistry and computational biology, typically from other fields. So I think it's more the rule rather than the exception that students, postdocs, and staff from the lab come from physics or math or computer science or chemistry backgrounds, and they haven't really done much work on RNA molecules or in biomedical research before starting the lab. And yeah, and that's a super fun thing.
It sounds like, I mean, you yourself have had a kind of multidisciplinary background. It sounds like the folks in your laboratory come from different disciplines as well. Could you speak to the strengths of that kind of diversity of training?
Yeah, I think, like in my case, I think there's a subset of, well, I'll just tell you my story. When I started my PhD, which is also at Stanford, in physics, I was pretty committed to working in theoretical physics and either in the fields of particle physics or cosmology. That's what I felt like I was best at and what I thought I would do. But after a few months, I couldn't find fundamental problems to work on where I thought that I could come up with an important and fundamental theory, while also having the prospect of having the theory tested in my lifetime. So even by that time, that was in the early 2000s, a lot of the frontier level questions in physics, whether it's what is the universe made of, what happened at the origin of the universe, we were creating experiments that took maybe 20 or 30 years to build, to send a telescope out into orbit or to build a particle accelerator. And it would take 40 or 50 years to get the experimental answer to whether you're right or wrong. And so I started looking into other fields. And one day, I was walking, I ran into a friend from college who was doing an MD PhD. And he said, hey, you should come and check out this talk. There's a cool lecture, and it's about a molecular structure called the ribosome, which I didn't really know what that was. But I went, and it was Peter Moore from Yale, a famous RNA scientist. Of course, I didn't know that. Presenting the work from his lab and Tom Steitz's lab on the first atomic resolution structure of a part of the ribosome, protein-making machine in all our cells. When I saw the talk, I just thought, how can like these strands of spaghetti arrange themselves into a machine that reads a message and produces a protein? It was like, it was like seeing a Ferrari or something self-assemble itself out of nuts and bolts. I just thought, how does that happen? So I figured that there would be pretty interesting physical questions, physics-like questions in that field of modeling RNA and RNA protein structure. And what really excited me was that you could do experiments. And so I actually spent the next few years in my PhD. I'd never done any experimental biochemistry before, but I was fortunate to be adopted by the Stanford Biochemistry Department, by Dan Hirschlag in particular. And they showed me the ropes. And even though I broke a lot of the glassware in the lab and burned through a lot of the expensive chemicals, and actually at some point set the lab on fire. They were very, the environment is so great here. It's so collaborative, and that I picked up those experimental skills, and I really loved it. I mean, I found that I love doing experimental biochemistry, even though I'm not very good at it. And so then after doing the postdoc, which is primarily computational work in the Baker lab, it's just been a joy now to be able to, do experiments. And sometimes personally, I'm in the lab right now, actually, running experiments. But then also to teach other folks who are, they're switching backgrounds. And so again, like maybe the majority of folks coming to the lab, they don't have experimental experience, but then, we have a culture where we teach each other those experiments. And I think Ewan might know some of the work that we've done in cryo-electron microscopy. That's been an awesome collaboration with Hua Chu, who's actually was in Houston and now is now here at Stanford. But the key people in the collaboration were students who were computational biophysicists, folks like Callie Capel, who in the last year of her PhD thesis said, I want to try something experimental. And she ended up demonstrating that you could apply RNA cryo-EM to really solve the structures of, in that case, about a dozen molecules that we had been previously intractable to us and the rest of the field. So I really, really relish getting to work with folks like that who have no fear and are coming from really distant fields. But also vice versa. I think it's been really, really nice to have people come into the lab who are more biochemically trained, maybe, or more cell biologists. And then nowadays, they're all learning how to do AI and how to train neural networks. And there are just these, key new ideas that happen when someone comes in with a lot of experience, but in a distinct field, and that hunger to answer questions or engineer new RNAs.
That's a great story. Really
Yeah.
wonderful story. Thanks for that, Riju. You started with physics, and at the moment we seem to be having a physics problem here in the studio. I hope it's not affecting the viewers back home, but the light on our teleprompter keeps going off and on. So just wanted to alert our guys in the camera crew over there that our teleprompter is misbehaving. Right now it's blacked out. So
Sorry,
I'll turn. Sometimes we can see you, sometimes we can't. But your voice is coming through great, and what a great story! Really loved it. And one of the things that you mentioned was your interest in actually having an impact, and your concern that maybe if you were doing fundamental physics, you wouldn't be able to get there in 40 years of work. And certainly you're having an impact right now with the switch that you made, applying physics to RNA biology. We are, I think, kindred spirits here, because we want to make fundamental insights as scientists, and that's not enough for us. We want to see those translated into something that's useful for people. So that's kind of the mission that drives our Center for RNA Therapeutics as well. What a great story. I'm going to turn to my colleague here, who's an expert in RNA origami, and see if you have anything to add about that discussion we just had with Riju.
I think if there's one question I could ask that's not going to be related to the talk, perhaps it's about your company, Inceptive. I'm just wondering, what your mission plan is there? What's your goal with that company?
That's right. So the goal of Inceptive, was founded during still while the COVID pandemic was raging on, and I'll tell you about a little bit of the work in the presentation here that we did on helping advance mRNA vaccines. I think for me, there was a frustration when, at the end of 2020 and by early 2021, our lab, our collaborators here at Stanford and at Pfizer we were making pretty rapid innovations that would improve the stability of mRNA vaccines and hopefully allow those mRNAs to get to more people around the world and help with the COVID-19 immunization campaigns. And we also felt like those technologies would help with future potential pandemics like from influenza. But it was also pretty frustrating, I think, to be doing this really interesting scientific work and then having these meetings, Zoom meetings, where both my lab and folks at Pfizer would be like, we're making these breakthroughs, but why aren't we working directly on the Pfizer-BioNTech COVID vaccine? We were working on model molecules. And the answer was, it's just not possible, or it wasn't possible, for universities to be directly working on products that would reach people, that go into clinical trials and reach people. That's more the exception rather than the rule. And so and around that time, that was early 2021, it was clear that, it wasn't clear when the pandemic was going to get resolved. There was a variant called the Delta variant that swept through India at that time, and then through the rest of the world, that just was absolutely deadly. It just many of the folks in my family in India were affected. I mean, basically everyone got COVID, in some cases with unfortunately fatal consequences. And I just felt like, very frustrated that the stuff that we were working on wasn't going out and reaching people. And so it was around that time that we started thinking about, ways to really get the technology out there into hopefully the next generation of RNA vaccines. And it seemed clear that, several things would have to happen. One is we'd have to raise some money. And there I thought back to a lot of the philanthropists who helped support our research in basic science and also on vaccines during the early stages of the pandemic. And I remember them all telling me, well, here's some money to help you support proof of concept. If you think you might try to commercialize the technology, I hope you can, call us back. We'd love to fund you at a higher level when you are trying to go to the next stage. And it was also important for us to really push on the AI methods. And so a lot of the work I'm going to tell you it looked like deep learning would be promising, but we wanted to work with the very best people in the world on deep learning. And that's hard to do in a university setting. We get to work with some of the best people, for maybe a year or two of their lives before they then go on to like a tech company. But I had been having conversations with, luminaries in the kind of AI field. Of course, they were at places like Google and Meta, and they said, well, if you start a company, we'd go and join you at a startup. That makes total sense. We want to be in direct. We want to be not just selling ads. We want to be working on products that can change the world, and so that was appealing to them. And I also found that people working with me, the biochemists at Stanford, also wanted to go and do something impactful. So that was in 2021. I was really fortunate to start interacting with someone who would eventually be the co-founder of Inceptive. Jakob Ushkareit is, I think, one of the really great and famous computer scientists in the world. He's one of the developers of this so-called transformer neural network, which is the base architecture for, ChatGPT and AlphaFold and other, world-changing AI. So Jakob, turned out had an interest in biology and was thinking about what to do, perhaps even outside Google, which is where he was at the time. So when we found each other and recognized our shared values and interests, then it became kind of a no-brainer to go and start the company Inceptive. So Inceptive's mission is to really develop RNA. We call it a biological software. We think of RNAs as having the function, mRNAs as having the function of encoding for proteins. So that's a kind of software, obviously, but RNAs can do much, much more. They can recognize their cell types. They can last for days or years. Actually, out in the environment, they can turn themselves off when they recognize small molecules, or turn themselves on. And these are all functions that nature can do, but we don't really know how to engineer them into new RNA molecules. And so we started Inceptive to go and figure out how to, just in a predictive way, design RNAs with that kind of exquisite functionality. And so that's been very exciting to then, to see that flourish over the last 2 or 3 years. I was, playing an important role in the founding, but very quickly, within a year, we amassed a critical mass of amazing people at the company. And now I've come back to doing basic science, while Inceptive is really bringing those design methods and approaches into the real world.
Spectacular story, and a really nice translation from the university into a company. And that's the typical route. Just parenthetically, we actually have the capacity here to generate products for folks, and we just got FDA approval last month for our personalized cancer vaccine program. And maybe there's some way to work with you to improve that. Well,
John, you and I have been talking, I think, since 2021 about about your program. So it's very exciting to hear about this FDA approval. Congratulations.
Thank you so much.
If you have interest in stabilizing those cancer RNA vaccines, perhaps either for shipment or in syringes, but also potentially in the human body, I guess the technology that I'm going to talk about maybe in some slides coming up Potentially we could explore that together. That would be a lot of fun.
Absolutely. Let's circle back on that.
Mm-hmm.
So maybe we should go ahead and get started with your presentation, Riju.
Great. Yeah, let me share my screen and I will get this going. So we will, oops.
I just wanted to mention that for our folks watching at home, you can join us with questions in two ways. One, you can text us, dial 37607 and text DeBakey, DEBAKEY, and your message. Then that'll get to us. Or you can go online to pollev.com and enter DeBakey, again, DEBAKEY, and you can just then text your question to us, and we'll ask Dr. Das. Go ahead, Riju.
Great. Thank you. So before I start, I will just remind you one more time that of a disclosure, I'm a co-founder of a company, Inceptive, that works in RNA design. Although I guess all the stuff I'll tell you about here will be from my academic work, the basic science work. So I'm going to start, as we already mentioned, with mRNA vaccines. I think we all know that in 2020, the first vaccines for COVID-19 were these mRNA vaccines, and they were remarkably effective, 95% effective against the first strains of COVID. Really miraculous medicines, but they were not perfect. And in fact, they did not allow for the world to be immunized. And that was for, I think, a pretty prosaic reason. It was these RNA molecules, or these RNA vaccines are not stable in refrigerators. And so it's hard to ship them in vials or prefilled syringes, outside or far away from their sites of manufacturing in the US and Europe. And this problem is, it's intrinsic to the vaccines being made out of RNA. There's a chemical issue where mRNAs are in some ways kind of suicidal. They're intrinsically unstable. And I'm illustrating that by showing you here a schematic, a cartoon of what an mRNA molecule might look like inside a syringe or in your body. It's not just a straight line as we often draw it. The RNA molecule will double back on itself to form these double-stranded regions, which I'm coloring blue here. But about half of the RNA will remain looped out or single-stranded. And these loopy bits can get into trouble. They can undergo a chemical reaction that's often called inline hydrolysis, where the RNA will attack itself at the backbone, and that can result in cuts over time, just for the RNA in solution. And even a single cut like this will render an mRNA molecule non-functional as a proper message in a vaccine. So actually, a little bit before the pandemic, we had been thinking about ways to get around this problem inspired by nature. Natural RNA molecules like the ribosome in your body actually form extensive structure. And so what if we could similarly take an RNA and make it, well, like all blue like this, make it so most of the RNA doubles back in itself to form double helices. We have to do that somehow while still retaining a sequence that can code for the desired protein. So this is both a really interesting problem, but also a very hard problem. Computationally, it requires for something like an RNA encoding the COVID spike protein, it requires a search through over 10 to the 600 sequences. So this is computationally quite difficult. And then it becomes even more difficult if you experimentally want these RNAs to be stable in a syringe or in a living cell. And so we turn to an approach that I think is quite powerful for these kinds of design problems, and that I hope that many more people in the field or in industry might turn to. And it's an approach that in our lab over the prior 10 years had helped us accelerate our RNA design research by up to 10 times. And it's a platform that's called Eterna. Eterna's tagline is played by humans, scored by nature. And it for, I guess, almost 13 or 14 years now, we have crowdsourced hard design problems in the form of this video game, and where people have, anyone on the internet can access to their phones or through a website, this interface where they can design yellow, blue, red, green symbols, which correspond to A, C, G, and U, and they try to make RNAs that will fold into the structure or functions that we are seeking, but haven't yet been able to figure out how to solve on our own or with our own supercomputers or AIs. And then these Eterna players can then share their sequences with each other, modify them, and improve them collectively. And then we've been taking their sequences and synthesizing actually every sequence that comes in through the video game. And we and our colleagues at Stanford and elsewhere have then invented ways to map these RNA structures and their functions to see if the RNAs have the properties that we desire. And through this virtuous cycle of online gameplay and experimental feedback, this Eterna community has produced an enormous number of scientific results, actually over 30 scientific publications. And many of these publications have been written not by me, but by these Eterna players themselves. When the pandemic rolled around, we decided to try to create a version of Eterna that for the first time would allow a design of mRNAs, RNAs that would code for proteins. I'm showing you here a little movie of someone playing a tutorial here where they learn about the genetic code, shown here on the left. And here they're going to design something, and there you see the bubbles. They were able to design a little tiny RNA that goes for a tiny peptide. So this, we started preparing it in late February when it looked like there was going to be a pandemic. And actually by March of 2020, which is around the same time that basically every university and company in the US shut down, we had this puzzle up and ready to go. And pretty rapidly, there were players who were able to make what looked like highly stable RNA structures for arbitrary, that coded for arbitrary protein sequences. But led by a player named Jill Townley and another player named Ome, this community got put together in a town hall, suggested that, hey, we don't have quite enough information to really design chemically stable RNAs. You really need to be telling us, well, if an RNA is going to form a loop, should it be one of these loops that's at the tip of a hairpin? Should it be one of these multi-loops? Is it better to have A's or U's in those loops? Are there special substructures that we should be designing to help stabilize an mRNA? And we and nobody else in the field knew the answer. So the players actually suggested that we then do experiments to go and figure out empirically what those rules for stabilizing an RNA chemically would be. And we said, okay, well, if you give us a bunch of fragments of designs of mRNAs that you would like to put into your full-length mRNA, we will figure out a way to go and synthesize each of those fragments and tell you how quickly they degrade or not. And so we launched what we call the Open Vaccine Roll Your Own Structure Challenge. And what I'm showing you here is just a kind of a map of all the different sequences that we got, in this case, from hundreds of different people on the internet. This is colored, the different colors and symbols are different people. And I'm just showing you here in each of these little clusters of sequences what some of the particular structures look like. So we have folks here who are creating these, kind of four-way cross-like motifs. We had players just giving us, strings of A's and U's and just really a whole gamut of different sequences and structures. And we didn't know how stable or not these would be. So to figure that out, we actually had to develop an experiment. Particularly Anne Cladwang and Hannah Wayment-Steele, working with me at the time, figured out how to take RNA sequences. We figured out how to synthesize them. There were companies who can make these molecules for us. We subjected them to degradation conditions where the RNAs would get cut up. Then we figured out ways to sequence which of the RNAs were getting cut up and where, and to compile statistics on where the hotspots were for these sequences and where they were protected from chemical degradation. And then using very simple machine learning, actually a so-called linear or ridge regression, we were able to take all of these data we had on roughly about 3,000 sequences. And we created a very simple model where we took the sequences that we had and the predicted secondary structures, carried out the simple machine learning, and we created a model called the DEG score model that let us take any new RNA sequence and try to predict its degradation rate at each position. We sum that up, and that gives us the degradation rate of the entire RNA or mRNA. And so this DEG score model, we then provided this to Eterna players, and they used it to then stabilize a bunch of molecules, including the COVID reporter, and we saw remarkable results. So here I'm showing you actually the degradation time profile for an RNA, this is an RNA designed with conventional codon optimization methods, like the ones that maybe, John, that you're using for your cancer vaccines. And it's degrading here under these conditions on the time scale of an hour. Whereas the RNAs that we call them superfolder mRNAs, the ones that the Eterna players were designing, lasted for much longer, at least 2 times longer than these original RNAs. And that so that was exciting for us. Even a doubling of lifetime is enough to make it so that you can, actually leave the vaccine out on the bench or on the fridge for months rather than weeks, or in this case, hours. The thing that you might be wondering is these RNAs that we designed to fold up quite a bit, do they still work as vaccines? Can they be recognized by our ribosomes in our cells, and can they print out proteins? And we were surprised to find that when we took these RNAs and with Maria Barna's lab, we put them inside human cells, actually some of them, most of them still produce protein, and actually some of them produce more protein than conventionally codon optimized mRNAs. So it seems like inside our cells, we have machinery that can actually pull apart these tightly packaged RNAs. And not only that, some of the RNAs that we designed to be more stable outside cells, or chemically stable, were also somehow more stable inside cells. And that allows then our cells to have more shots on goal of printing proteins from the RNAs. So we end up with more protein. The real, I think, test of this was with our collaborators at Pfizer Viral Vaccines. They took these RNA sequences and they measured the protein expression activity for some of these RNAs. And what you'll see here in green this is an Eterna player's design called Bugakman's Lost LD. And it's producing as much protein as the next best sequence that we had, which came from a commercial codon optimization method. More than that, when they left this RNA formulated in lipid nanoparticles on the bench, or actually they put it at, I think, at 42 degrees for 2 weeks, the RNA was still functional after 2 weeks, whereas these other designs started to lose their activity. And so that's led to this paper, which I'm really, really proud of, which you can grab. Catherine Lepik's the first author. The thing I want to highlight here, which I think is unprecedented, is that this paper has many different authors. It includes, I think, a couple of hundred folks from the Eterna Massive Open Laboratory, and it also includes 9 scientists here from Pfizer Vaccine Research and Development, which at that time was the biggest drug company in the world. So I think this is a kind of a first for this community science type of approach. So since then, we were curious to see whether we could do better than this DEG score model. As I mentioned, the DEG score is a very simple machine learning. It's a kind of approach that we and others would be able to apply even back in the 1980s or 1990s. What if we could apply these modern AI deep learning methods to the same problem? Could we do even better? So we got a taste of that through yet another academic collaboration, where we turned to, we have wonderful people at Stanford and through our collaborators who are developing machine learning methods, but we turned to another crowdsourcing platform beyond Eterna. It's called Kaggle. And so some of you may have seen this. This platform involves now well over 1 million users. It's actually now part of Google. And you or anybody on the internet, if you have an interest, can take part in competitions to develop AIs, to do data science, to predict the financial markets, to help folks develop new protein drugs. And in this case, we were going to do it, help stabilize mRNA vaccines. And Kaggle is really wonderful because although it's a set of competitions, there's a really wonderful community who help each other to get better at data science and through these competitions. And even my older daughter, she's learned Python and basic data science by taking part in these tutorials online. So we turned to Kaggle in 2020 with a competition called the Open Vaccine Competition. And what I just want to point out is that for actually not very much prize money, we were able to recruit over 1,600 independent teams. So I think this involved so many thousand actually unique people to come and work on this mRNA stabilization problem. And what we decided to do was to ask them to do better than we did on the DEG score in taking our data that we'd collected in the Open Vaccine Roller Run Structure Challenge, see if they could do better. I'm just going to cut a long story short by showing you just the final results here. We scored different Kaggle predictors based on how well they agreed with data that we had kept hidden, actually that we hadn't even collected at the beginning of the competition. And lower is better, meaning tighter agreement. And so this is our DEG score up here as the red score, and these are two different actually test sets. And black is the best you could conceivably do given our experimental noise. And the Kagglers actually got us more than twofold of the way better to being perfect here in a competition that took only 3 weeks. Quite remarkably, the key event that occurred was a small set of the Kagglers created what they call a shared kernel, a shared notebook, and then all the best predictors then iterated on this notebook and tried a bunch of different deep learning approaches, then got them to the very, very best. So for now, I'm not going to go into the details of these AI methods, but I'm happy to take questions on them. I did want to point out that we had done secretly, we had also planned to do a separate rigorous test of these models. Rather than just looking at how well they did on our test set with some number, we actually took a bunch of other sequences, full-length mRNA sequences rather than fragments, had measured their degradation rates. And we checked to see how all the various predictors we had, ranging from DEG score up to these are the two top Kaggle predictors, how well they predicted those data. And again, remember, the Kagglers, they didn't have these data on full mRNA sequences. In fact, they didn't even know that we were collecting them. And yet, maybe you can just look at the numbers here. Here, higher is better. These Kaggle models did significantly better, actually a factor of 2 better than the original physics-based model that that I and people in my lab had published early in the pandemic. So we got quite excited about this. This is a separate paper called Deep Learning Models for Predicting RNA Degradation. And this is some of the work that also helped convince us that further scale up of the AI approaches, as well as further scale up of the measurements, might benefit from commercialization in a company like Inceptive. But we did notice that there are still some misses with this level of data. So I'm going to show you an example. This is a little RNA fragment. It's highly AU rich for the RNA aficionados. Typically, AU pairs are not that stable. Where the Kaggle first place model predicted that this RNA should fold up really well. And although it'll be degraded, those are these orange residues that are at these loops, at the AU base pair that predicts that those are not going to be degraded. However, when we actually did the experiments, those predictions turned out not to be true, even in the top place models from the Kaggle competition. And our experiments show that under the same conditions, this RNA actually is going to get degraded. Apparently, these stems open up and can be hydrolyzed. So this motivated us to potentially start thinking about scaling up our data generation. We had generated data on about 3,000 sequences. The number of data points you can access on the Kaggle Open Vaccine competition was about 300,000 data, but that's actually kind of small on the scale of what we need to do for what deep learning needs to do for getting maximal accuracy. So we started thinking about whether we could scale up to maybe by a factor of 100 or 1,000 to 300 millions of data points, or maybe even billions of data points. And to do that, we went back to our protocol for mapping chemical degradation. We took advantage of some other pieces of protocols that let us get information on RNA structure. There's something called chemical mapping methods. Here, there are reagents that instead of degrading the RNA will actually put little tags on the RNA. And it turns out when you have that situation, there are tricks that our lab and other labs have developed for rapidly getting data on how chemically reactive all the different tags were. But then these methods have the potential for going beyond thousands of RNAs up to potentially hundreds of thousands or millions of RNAs. And in fact, we went back into the lab, this is back in 2023, and showed that indeed, if we get 1 million sequences synthesized, so almost 1,000-fold more than what we had before, we can get very high quality data. Each of these rows corresponds to a different RNA sequence that we've synthesized. And the black or the white values tell you how chemically reactive or not each of those positions are on each of the sequences. So, and this is 1/1000th of one of the data sets that we were collecting. When we have independent experimenters do these experiments, actually with molecules that were synthesized by different commercial vendors, we get outstanding reproducibility. So if you just, so this seemed like a really powerful method for getting data for training AI models. So we were then in a situation where we could get measurements on 1 million diverse sequences that were sensitive to structure, but we had a question of how do we get 1 million diverse sequences to go map? After all, we probably shouldn't be looking at the tRNAs over and over again. That's not diverse enough to give us information to train an AI. And also, we weren't sure how we could get this AI to be trained that could go and ingest these kinds of data and then later be able to predict structures of RNAs. So for both of these questions, you might guess what I'm going to say next. Our solution involved turning to the internet and crowdsourcing again. So just as we had taken advantage of these two Eterna and Kaggle crowdsourcing platforms in our prior work, we decided to revive these projects on Eterna and Kaggle in the context of now scaling up what we're doing to the scale need for modern AI. So on Eterna, we set up a set of challenges. They're called the OpenNOT challenges. They started last year, where we asked in each round for Eterna players to generate, in this case, 750,000 designs. And this is a screenshot of them, producing tons and tons of designs for us of RNAs that would form a wide variety of structures. And then we initiated on Kaggle what we call the Stanford RibonanzaNet RNA folding challenge. The data set is called the RibonanzaNet data set. And as you'll see, the resulting models are also have that tagline as well. So we launched this about a year ago. We launched the RibonanzaNet competition on Kaggle. We had about data on 1 million RNA sequences at the beginning. For the aficionados, we had both shape and DMS profiles. We had these came from many different sources, including synthetic RNA origamis, like the ones that Ewan designs. But the plurality of these sequences came from the Eterna platform. And what we collected during the competition were 1 million new RNA sequences, actually all with longer lengths, to help us evaluate whether the Kaggle models were actually predictive of novel RNA sequences. And here I'm showing, again, the scores of these Kaggle models on 2 different test sets. Lower is better. And our baseline, which we call the RNA degformer, this had arisen actually out of the prior Kaggle competition, is here. But you can see that there are actually 20 models that do better. They're lower and to the left on this diagram coming out of this new Kaggle competition. And in fact, if we took the average of the models of the top 3, we do even better than anything that was submitted in the competition. Especially there were tons of interesting, diverse ideas that were explored in the Kaggle competition that did better than what we could come up with in-house. In fact, we had an embarrassment of riches. We got too many models. Again, for the sake of time, I'm not going to go into the details of these neural network architectures, but I can tell you they were all variants, but interesting variants of the transformer architecture. So we actually, for those of you who might want to do this yourselves, set up your own Kaggle competition, you would be faced with the same conundrum of how do you deal with all of these different machine learning models? How can you use this in your research in the future? The trick that we developed is actually, we asked each of these competitors to make a whole bunch of predictions. Then we use those predictions to train a single final model that was a kind of student model. And we call that one RibonanzaNet. So RibonanzaNet is a model that can go from sequence to predicting chemical mapping data. It's freely available on the internet and through Kaggle. You can also access all the raw data that is used to train it. It was developed by Shujun He, who's actually a graduate student not too far from you at Texas A&M, who we met through our first Kaggle competition. And he just pivoted his lab at Texas A&M to working on mRNA design. They're doing brilliant work. And Shujun actually stayed on Kaggle as a grandmaster. And then instead of taking part in the RibonanzaNet competition, he decided to switch sides and host the competition with us, and also to train this final model. And with Shujun's help, we actually revisited the Kaggle Open Vaccine competition with this RibonanzaNet. And now, pre-trained on this much bigger data set and then fine-tuned on the same data that were available in Open Vaccine, I'm showing you here the results of those prior models. And then here's RibonanzaNet, lower is better. It's outperforming all of the prior models from, in that case, 1,600 teams substantially, and is doing it because it's learning from all these additional data that we've collected. And what I love is, like, even for these misses in the prior RibonanzaNet competition, this is that RNA that actually gets degraded, although the prior Kaggle competition, those models did not predict the degradation. This RibonanzaNet model actually understands that this RNA actually does unfold when you put it in a test tube, and gets hydrolyzed. And so it's doing a much better job of capturing these, even qualitatively capturing these experimental data. Okay, so I'm actually going to stop there. Let me just quickly summarize. I've been telling you about a new approach, or a distinct approach, let's say, a conventional approach, to understanding RNA structure and function and maybe designing therapeutics through a set of worldwide competitions. First, I told you about how Ceterna video game platform had an open vaccine challenge that helped reveal principles for designing superfolder mRNAs. And then those molecules, both in our hands and in the hands of our collaborators at Pfizer, led to increased stability of the RNAs during storage, but also led to sustained, or in some cases, increased protein expression. Those are based on collecting lots of data and developing a pretty simple machine learning algorithm based on Eterna designs. We then reached out to a different platform, the Kaggle platform, to develop even more sophisticated deep learning algorithms. And that has led to the state-of-the-art in predicting RNA degradation. And then we have now, just in the last few months, have run another Kaggle competition called the RibonanzaNet competition. It's resulted in a neural network called RibonanzaNet, which has even further improvements. And these have come from training from about 1,000 times more data that we've collected as part of this RibonanzaNet competition. So with that, I want to really thank the people who've been doing this work with me. On open vaccine, it was an all-hands effort. Included Hannah Dosun and Christian from my lab, and then Maria, Gunn, Craig, and Catherine from the Barna lab. Pretty much all of these folks are now either professors or they founded companies working in mRNA design. Some of them are actually at Inceptive. I also want to thank for Ribonanza everyone who's been in my lab for the last year and a half, especially Thomas, who's been helping with the Kaggle competitions, and then especially Shujun, who's been, and Ray, who collected the data, Ray Huang, and then Shujun, who's been, was both a Kaggle Grandmaster and RNA graduate student, and in this case, the host of this new Kaggle competition. These are the top 10 Kagglers who have helped write the paper with us. These have been our funding sources. I want to thank everyone on Eterna, including these player developers who are now actually the leaders of Eterna. They're all former players. And I want to thank the many people in the community who've contributed, especially to open vaccine. I want to highlight here Alexis, who's also based in Houston. She finished her undergraduate degree at Prairie View A&M with Dr. Gloria Regisford, and was one of the star players in the open vaccine competition. In fact, she about a year ago won an award from Ebony magazine called the HBCU STEM Queen Award. If you want to read more about our work, it's available. The Ribonanza stuff is in a preprint. And if you want to take part, I think this gets at what John and I were talking about right before. We're continuing to develop mRNAs, both through Inceptive in a commercial setting, but also if you want really good RNAs that have been developed with feedback from humans like me and others who've been thinking about RNA for a long time, Eterna is also continuing to run vaccine design challenges. We just finished up last month a competition to design COVID mRNA vaccines that would go into the clinic. Here we provided this new neural net, RibonanzaNet DEG, as one of the predictors in the game. And if these are successful, these designs that have just come up are being tested by our partner in PopVax, and they'll go into the NIH's Project NextGen trials, which are going to be held next year. So this was just announced last week. So we're crossing our fingers. That'll be, I think, the first time that molecules from a video game are going to go out into clinical trials. And then if you're interested in developing shelf-stable mRNA therapeutics or vaccines, they could be personalized cancer vaccines. If you are willing for the RNAs to be available on a nonprofit basis, then just email me and we can set this up on Eterna and to use this Ribonanza DEG predictor. And then if you are maybe not an RNA medicine developer, but if you're just someone who's interested in helping contribute to the development of RNA medicines, if you're a student, maybe you're a retiree, then you might, instead of solving crossword puzzles or Sudokus, you might want to join Eterna. This is the URL. It's easiest to play probably through a web browser, eternagame.org. And we currently have this really fun lab where we're developing T-loops that we would use for designing 3D RNA nanostructures like the ones that Ewan is developing. And these would potentially go into our future RNA vaccines and other RNA therapeutics. All right, so I'm going to stop there and happy to take questions, John, Ewan, and from the audience, from people who are texting in.
Thank you, Rijo. Spectacular, innovative, exciting, fun. We do have questions coming in from the audience.
Great.
Do you want to ask the questions for Rijo?
Yeah, actually, I have a burning question of my own that
Sure,
I
go
would
right
like
ahead.
to ask as well. One thing you didn't touch on here is modified nucleobases. As you know,
Right.
lots of mRNA medicines utilize things like methyl pseudouridine, 5-methylcytosine, and these sorts of things to reduce immunogenicity. And although they don't disrupt the Watson-Crick base pairing, they presumably alter the thermodynamic landscape. And I'm wondering how you expect this might affect your structure predictions, or are you going to have to do a million modified RNAs in your next data set, or what your thoughts are on that?
Yeah, so a couple of things here. So, right, so almost all the COVID vaccines that were approved for mRNA used pseudouridine or M1 pseudouridine, and of course we knew that pretty early on in the pandemic that Moderna and Pfizer and BioNTech were going to be using these modified nucleosides. So we checked that the superfolder mRNAs, we checked their stability and their protein expression with pseudouridine and M1 pseudouridine. And we were worried that thermodynamic landscape would be different, and so these RNAs, all of our design principles for stabilizing the RNAs would just go out the window. Surprisingly, all the RNAs did better with the modified nucleosides. And they did better for a number of reasons, but the one that was most surprising to us is that if you have a pseudouridine instead of a U, that base appears to become less nucleophilic, and so it's less likely to cleave itself. So there's an unexpected chemical reason. Some of this is also structural. So what we would, in our current studies, we would like to be able to just model the RNAs folding with pseudouridine and 1-methylpseudouridine. And I actually did an experiment earlier this summer where I just took our existing RibonanzaNet libraries and remade them with pseudouridine and 1-methylpseudouridine. And we can get beautiful data on those RNAs with the different nucleosides. And those data appear to allow us to then fine-tune our model to predict what happens if you, instead of having U, you have the modified bases. So actually, I think it's actually a very small lift to go and update RibonanzaNet to predictively model basically any modified nucleosides. You don't have to go and redo everything. You can just, in fact, just redo the experiments on the RNAs we have sitting in our freezer with modified nucleosides. It's fairly straightforward. So if you guys have interest in that, or if other people on the call who are watching this have interest in that, doing collaborative research on the modified nucleosides, we'd love to do it. I think it's really pretty straightforward.
Great, thanks for your nice answer.
Some other questions coming in.
Yeah, so we've got a slew of questions about Eterna. It looks like there's some excitement about trying that out. So this is a video game. What console is it on? What's the objective of the game?
So it's a game that you can play in a web browser. There's also an app on your iPhone. And when you first go in, you don't even have to set up a login. You can just go to this website, hit play, and you can see what it's about. Most of the initial problems, you are tasked with trying to design an RNA sequence that will fold up into like some snowflake-like structure. And it's a purely two-dimensional game, so it's really, I don't know, it feels a bit like, it does feel like a Sudoku or something, but it's a molecular Sudoku. And there are tricks. As you start changing the sequence, you think I'm gonna get the A's to pair with U's and the C's to pair with G's. More often than not, you'll make your sequence and it'll find a misfolded sequence. So we have a computational folding engine in the game that's trying to predict what will happen in real life. And more often than not, when you start making the sequences, there's some tricks to stabilize just the structure you want. So that's the game. And once you've played about 20 or 30 of those puzzles, you then gain admission into what we call lab puzzles. And then you jump right in and you start designing structures, or in some cases, RNAs that have multiple structures, or RNAs that have weird so-called pseudoknotted structures, or T-loops, like what we have here, where we actually don't quite yet know how to accurately simulate the folding of those structures. So we're asking you to generate as many sequences as you can. Then we do experiments, meaning my lab and colleagues do experiments, and then you get way more points if you actually design something that works in real life, in the test tube, or actually when we test them inside living cells. So so that's the overall gameplay. There's something that you can do kind of on a daily basis or weekly basis. You solve little puzzles. And then there's this broader game of actually doing science, where you get back at some point, a big component of your score becomes how well you do in actual so-called lab challenges.
Great.
And I think there's still some other questions.
Yeah, a couple more questions. Someone asks, are there any advantages to modeling the experiments in 3D space or using 3D printing, for example?
Right, yeah. So for computer modeling of the RNAs in 3D, we would love for Eterna or for any of these design efforts to really visualize the RNAs in 3D. We have found that for most of the problems that we care about, there are some AIs that try to predict RNAs in 3D. There's a variant of this AlphaFold predictor for proteins that can make predictions for RNA, but they do a terrible job with the sequences that we've looked at. My lab also, we invest a lot of effort into solving 3D structures of RNAs using a method called electron microscopy. And we found that whenever we make predictions, they're just not very good for the RNAs that we've been looking at. So we'd love to include 3D modeling as part of Eterna and just part of our modeling pipeline, but it doesn't seem like we're there yet as a field. Although a major goal of ours is to actually use the data we're collecting on Eterna and then go back and improve those 3D modeling AI algorithms. In terms of 3D printing, I've got a bunch of 3D printed RNAs where we solve RNA structures experimentally, and then we look at them in 3 dimensions, either using 3D printing or using VR glasses. And I learn a lot from those models. Although we still haven't quite figured out how to get that scaled up to everyone who's playing Eterna. So if you have ideas there, we'd love to hear them. Maybe we should give out as prizes 3D printed little sculptures of people's molecules.
Riju, it's really a brilliant approach and a lot of fun, just really innovative. I just wonder though, it doesn't take into account RNA binding proteins and how the structure may affect the interaction with RNA binding proteins that could stabilize or reduce the stability of the RNA. What are your thoughts about that? Is there at some point can you incorporate RNA binding proteins into your models?
Yeah, I think that's yeah. We have, I would say, half of my lab right now is working on that. It's a hard problem. It's historically been a very hard problem to model RNA protein interactions from scratch, much less design new ones. I can design a CRISPR therapeutic for gene editing, but really the protein RNA interactions in there we're copying what nature has already done, and the RNA interactions are just straight Watson-Crick-Franklin stems. So there's a little code that we can use. But for what a lot of us want to do is we want to create an RNA that when it goes inside your cells, it recruits the ribosome, or it detects what cell type it's in based on the proteins that are in there. And it's been a kind of holy grail problem in our field to model and design RNA protein interactions. So I have to say, my lab and no one has been able to predict those. Right now, there's a competition called the CASP competition, Critical Assessment of Structure Prediction competition. Or I should say, it happened over the summer, and we had 30 or 40 prediction targets that were RNA protein structures. And actually, my lab helped organize and assess that category. And we will announce in 2 weeks at the CASP conference how well predictors have done in modeling those interactions. So I guess we'll keep our fingers crossed to see if there's been a major improvement there. If there hasn't, then we're taking one of these, like a big data approach, and using Eterna and Kaggle. I think that'll be potentially a way forward for RNA protein interactions.
Well, really spectacular work, all the way from physics to chemistry to biology, and having an impact on human life. It's very encouraging. Thank you so much, Riju, for your talk today. Thank you for spending some time with us. And I will circle back with you about potential interactions with our personalized cancer vaccine program. Well, thank you so much. Thank you so much for joining us on the show today. Thank you, Dr. McCrae.
Thanks, John.
And keep up the great work. Well, thank you all for joining us today. You're at the cutting edge of RNA therapeutics with Drs. Cooke and McCrae here, and Riju Das was our guest today. Thanks again, Dr. Das, for a spectacular presentation.